So the last talk is on the chemistry prize. And as you'll see, there's certainly some interesting connections to chemistry, but the applications are in biology, or especially what I think has made this prize important. Jeff Kaplan is going to give the talk. He's the director of our bioimaging center located at the Delaware Biotechnology Institute. Jeff earned his PhD at Yale University in the Department of Molecular, Cellular, and Developmental Biology. After graduating, he stayed on for an additional 1.5 years as a postdoc at Yale, working in their Center for Genomics and Proteomics, because he had decided he actually wanted to learn how to run a core facility. That's not many people see that as a career path when they're getting their PhDs, but Jeff did identify that as something he wanted to do, and we're very lucky to have him here directing our bioimaging core facility. He also, of course, has his own research program. He's been involved in using microscopy to study innate immunity in plants. And he's running a research group here that focuses on characterizing organellar dynamics using live cell imaging and the development also of novel imaging techniques. The prize, as he explained to me, is actually for an older technique that has been superseded by some newer developments. And he's going to talk about some of the work that is going on here at UD, as well as giving us some background on the work that won the prize. So Jeff?
Okay, Doug, thank you very much. So it really is my pleasure and honor to describe to you the Nobel Prize in Chemistry for the development of super-resolved fluorescent microscopy. And it was awarded to these three men here, Stefan Hell, William Warner, and Eric Bexit, for two different techniques for bypassing the diffraction limit of light microscopy and developing super-resolution techniques. Stefan Hell developed a technique called STED, which I'll explain, and William Warner and Eric Bexit worked together to develop a different technique called PALM. Over the course of the talk, I'll explain what those two techniques are, and I'll give you a history behind their path to discovering these two methods. I'm going to first start very simply to talk about size and resolution. And these things obviously can be seen with your eyes, and I challenge you to look next to the person next to you and look at their hair. You can obviously resolve that with your eyes. But then I challenge you to look next to them at their skin and try to resolve their individual cells on their skin, or the bacteria, 1,000 different species of bacteria that generally roam our skin, or the mitochondria, which are the organelles within a mammalian cell. And for that, you generally need a light microscope at the very least to resolve these various things. It doesn't mean you can't look at these other things with a light microscope. This is actually a picture of hair that I took with Colin Thorpe, the professor here at UD. This is actually Colin Thorpe's hair. We are now developing some super resolution applications. So you can see the amazing detail, even from a standard light microscope, that you can get from hair. However, if you want to resolve things smaller than 200 nanometers, and each one of these points on this is a fold lower in size, you can, because there's something called this Abbe diffraction limit, and it was defined by Ernest Abbe in 1873. And that prevents us from resolving things smaller than 200 nanometers. It's based on the wave properties of light. Light diffracts. So if you want to resolve things smaller, such as viruses, proteins, and small molecules, you really need electron microscopy. And indeed, we can look at things on a full range of this size scale with electron microscopy. Here's an ant. That's actually the head of the ant. And these are virus-like particles on a scanning and an electron or transmission electron microscope taken at the bioimaging center here. But there's a reason why we don't always use an electron microscope. First of all, sometimes you're just looking too close. You wouldn't read a billboard with a magnifying glass. The other reason is that to use an electron microscope, you often have to kill the sample. So if you're looking at any live cell process, you can't, and you want to look at something that's smaller than 200 nanometers, you wouldn't want to use an electron microscope. And that's really what the award was for, was breaking this diffraction limit and creating these super resolution techniques which allowed you to then look at live cell processes with light microscopy. Okay, and I'll talk about the two different ways in which that was done. Okay, the first one was done by Stefan Hell. And he was actually obsessed with breaking the diffraction limit. He recognized this as one of the real grand challenges of his field at the time. And you know, when he was a senior scientist in Germany, his colleagues told him, you know, you can't argue with the laws of physics. You know, this is a law that was written well back in the 1800s, 1873. But he pursued it anyway. And in 1993, he actually was reading this paper on stimulated emission depletion, which was, he said, a concrete concept that he could pursue, and published a paper then in 1994 in Dr. Wickman's lab explaining how this could break through that diffraction limit barrier. 6 years later, he was able to actually build that microscope at the Max Planck Institute and demonstrated that technique in live cells. So let me briefly explain the problem here that he solved. So I'm going to very crudely depict a fluorescent molecule as this ball and stick diagram here. But for fluorescence to work, you would hit a fluorescent molecule with one wavelength of light, and then it would fluoresce at a longer wavelength of light. So here, hitting with green light and fluorescent red light. The problem is, because of the diffraction limit of light, this molecule, which is a couple nanometers in size, will actually look like 200 nanometers or larger. So if you were trying to resolve something that was smaller than 200 nanometers, let's say we were trying to spell out the letters UD with fluorescent molecules, what would happen is when you excited all the fluorescent molecules, they would turn on like this. And this is what you would see. You wouldn't be able to actually resolve the two letters. So the idea that Stefan Hell came up with was to use this technique called stimulated emission depletion. And what that does is, using high energy, it can actually suppress the light or turn off the light from molecules. And so you normally excite it again, and all the molecules would turn on. But if you used another laser beam, the shape of a donut, you can actually turn off all these other molecules, and so that you actually see light only from the molecule in the center of that donut. Okay. And then you can then scan across the image like so, turning on and turning off molecules and detecting the fluorescence on a smaller spot. And that's basically how we got higher resolution. Here's a real example. Well, here's the applet created by Mike Davison of the endoplasmic reticulum in mammalian cells. And this is what would be a wide field or normal fluorescent image of the ER. And here is a super resolution image of that. So the detail you get out of it is pretty remarkable. This is actually from one of Stefan Hell's papers showing the resolution enhancement of the ER compared to a confocal microscope. And just to connect this to the Nobel Prize in Physiology and Medicine, this is using instead on live cells of neurons, this hippocampal slice taken from a mouse brain, and they were looking at the dendritic spines along that neuron. So the applications are incredible. Here we're looking at a single neuron, so it's pretty amazing what you can see with that level of resolution. And now this instrument is used all over the world to study really a wide variety of biological questions. So the second method, and I'm just going to jump right into this, was instead of turning off molecules, we basically look at one molecule at a time. So we're still using diffraction limited light here. Okay, we hit it with green light, it fluoresces red. But if you look at one molecule at a time, you can just Gaussian fit the center of that molecule, and then you know the precise location of that molecule. So what you do is look at single molecules at a time, and then you can resolve what you're interested in. But the key to that was actually being able to detect single molecules. And that's where William Warner came in. So in 1989, he was the first person to measure light from a single molecule. And just a bit of history here, John Raybould here at UD actually was at the IBM research lab at the same time as William Warner. And he told me, described him as a very, very quiet personality, but he was extremely passionate about science, extremely focused, and he had a vision of where he wanted to take his work. So he went on and he basically created the foundation for work by others to detect single molecules at ambient temperatures in liquids. In 1997, John Raybould actually came here to start the Department of Material Science and Engineering. And William Warner went to UC San Diego, where he met another Nobel Prize laureate, Roger Shen, who was working on fluorescent proteins and creating different color fluorescent proteins. He actually took one of them, the green fluorescent protein, showed that it can blink, basically turn on and off, and if it got stuck in what's known as a dark state, you can then reactivate it and turn it on again. And so this was a really important piece for Eric Beksic to do his work on creating that single molecule super resolution method. So Eric Beksic was hired in Bell Labs in 1989. He worked on another super resolution technique to break that diffraction known as near field scanning optical microscopy. And he actually left science for about 7 years. So he was kind of fed up with academic science. He decided, I don't want to do this anymore, I'm done. So he kind of left. But right around the same time, he published a paper that explained the single molecule localization technique. And just like Stefan held, he was actually completely obsessed with breaking the diffraction limit of light. And so he also recognized this as one of those grand challenges. And so at one point he met with Mike Davidson, and just like the Nobel Prize in Physics, there are often a lot of people involved in the work. And he met with Mike Davidson, who had these fluorescent proteins from Jennifer Lippincott-Schwartz. And Jennifer Lippincott-Schwartz made these fluorescent proteins based on the work from W. Warner. And these proteins, if you hit it with a UV light, will actually fluoresce green. And so you could turn on, they're normally dark, and you could turn on individual molecules at a handful at a time. And then if you image it for long enough, those molecules will bleach out, and you can turn on another set of molecules. Yeah. And so after hearing that, that was kind of like a eureka moment for Eric Beksic, and he contacted his friend at Harold Hess, who he met at Bell Labs, one of his colleagues, and he started to build a microscope in Harold Hess's living room. So as you know, many companies have started in a garage. Well, they started their company in a living room, which when you think about it, is much smarter, right? You're building a microscope, you don't want to do it in a garage. And I'm just going to explain the technique one more time here. And so this is basically what they built. They had a lens here that could turn on single fluorescent proteins at a time. And as long as they were 200 nanometers apart, they can then accurately determine their location, much smaller than the diffraction limit of light, and then take hundreds of thousands of images and combine those images to create a final high-resolution, super-resolution image. And he called that technique photoactivated localization microscopy, or PALM. Here's the work from that original paper. And I remember as a grad student reading this paper, I was actually analyzing mass spec data at the time, and my PI came up to me and said, oh, you have to see this paper. And I'm like, oh, I'm busy. But after reading it, this is one of the papers that actually convinced me to go into the field of microscopy. And the resolution enhancement was phenomenal, and it's very easy to see. This is a normal light microscopy image, and this is a super resolution image. And if you look closer, you can see the single molecule localizations here. He then went on a couple years later to show it in live cells at focal adhesions. And he sold the licensing rights to a company called Zeiss. And it's actually quite important to commercialize these technologies because, yes, it has great impact in those individual research labs, but when you commercialize it, you make it available to everyone. And so we're actually lucky enough to have one of these instruments here at the University of Delaware with support from biological sciences, INBRI, COBRI, EPSCoR, a lot of these institution-wide support mechanisms. We're able to bring this microscope here. And this is real data coming off of the instrument. This is what it looks like. And it's hard to see, but there's the membrane here of a fungal tip. And then there's this area here called the Spitzenkorper that we're able to resolve at higher resolution. This image was created with 8,000 images. So we took 8,000 of these images. We're localizing these single molecules to create the image here. And it must be very, very hard for these committees to decide who to award the Nobel Prize to. And just like the Nobel Prize in physics, there are a lot of other people that would certainly worthy as well. That includes Matt Gustafson, who developed another technique called structural illumination microscopy, and Xiaowei Zhang, who created a technique called stochastic optical reconstruction microscopy. And we can do both of these techniques on this instrument as well. Here's an example of structural illumination microscopy that gives you, it's only a doubling of resolution, but it's pretty remarkable what you can get with just a doubling of resolution. Here's an example of platelets. That's just a doubling of resolution. The advantage of this method is it's very easy to look at multiple colors at the same time. It's much easier than some of the other methods. We're now actually doing this in combination with electron microscopy. So we can take super resolution, this is a super resolution image taken on our instrument and then brought over to Chao Yingni's facility here in the ICE lab, where we took a scanning electron microscope image and then overlaid. This is in collaboration with Donna Wolf. And we could do the other technique called D-storm on platelets to look at the microparticles as well. And so I'm going to leave with a definition of innovation, which is creating or uniquely applying new tools to solve old problems. And these scientists for sure did that. They created these new tools to solve a very old problem with the diffraction limit of light. And now we have the opportunity to uniquely apply that new tool and to solve questions that were previously unsolvable. And this is just an example of the impact that this is having on research. These are the publications that have used super resolution over this pretty short time period. Now we have over 20 different research groups using our super resolution microscope to advance their research, which is pretty impressive. So I'm going to leave with just saying that this is really inspiring innovation, and we're pioneers in the field of super resolution nanoscopy, and they're still continuing their work, and it's really exciting. Stefan Hell is creating techniques such as Resolve, which get over some of the phototoxicity effects of super resolution instead. Eric Bessig just published a paper on lattice light sheet microscopy, and so they're still creating incredible, innovative ways to break the diffraction limit. And so it was really, and this is a direct quote from one of the Nobel Prize committee members, is this development is expected to revolutionize biology and medicine by, not the least, eventually allowing for realistic quantitative descriptions at nanoscale resolution of the dynamics of complex, multidimensional, molecular biological processes that define all phenotypes of life forms. So it's a fairly new technology, super resolution, but I think it's going to have really a profound impact on the biological sciences in the near future. And with that, I'd be happy to take any questions.
A couple questions in back there. So.
hi, Jeff. So when I saw this announcement, I was actually a little bit surprised that it came out as a chemistry prize rather than a medicine prize. So I'd like you to discuss a little bit about why this is a chemistry prize and the chemistry aspect of these different techniques.
That is a very good question. It can be argued that this, and some people thought maybe the two should have been switched, right? The chemistry part is probably William Warner's work, who did a lot of chemistry work looking at single molecule fluorescence. It's hard to argue that, you know, the work done by Stefan Hell and the work done by Eric Bessick really was more physics than chemistry. And really, the argument is that, you know, everything's becoming multidisciplinary. And, you know, this is work by two physicists and a chemist for the Nobel Prize in Chemistry, showing the application of something in the biological sciences. And they really got the award for the applications in live cell imaging. There's other people that didn't get the award because the rationale was that they're much harder to implement in living cells.
Speaking as a chemist myself, I'll say that, you know, that when these guys were starting to do it, this was a really hot area in chemistry. There were just, you know, where the line is between physics and chemistry is not so clear. But I don't think when they started it, doing the sort of, you know, being able to detect fluorescence from single molecules, nobody was thinking about biological applications at that time. Another question back there.
Jeff, very nice presentation. So here you show that the wonderful super resolution images. As I understand, all those images has to be taken from a fixed tissue and for a long time. So basically we are trading time with spatial resolution. So I don't know, is it possible really we can follow the dynamics of very complex system at such high resolution? Do you think the future direction will lead us there, or we are still fundamentally balanced temporal and spatial resolution in this field?
That is always going to be a challenge, and microscopy is balancing temporal resolution, right, physical resolution, and also phototoxicity. So Eric Bessick actually admits that PALM is very difficult to do in live cell imaging, and really one of the few applications is the study of focal adhesions, which he published back in 2008, and he'll admit to you that he hasn't done much more with it since then because of some of those limitations. That being said, he just developed a new technique that gets around some of those limitations, lattice light sheet that can be used for super resolution, where you can track take a lot of high resolution 3D volumes of cells very rapidly, and they're able to track single molecules using PALM for example, microtubules extending. So I think as time moves on, the same thing with STED. STED had the same problem, right? You had to scan very slowly and with really high laser power, and you were basically cooking your sample. These new technique called Resolve that Stefan Hell also developed uses reversibly photoswitchable proteins, which you use 10 to the 6 less laser power. So I think the field is going to continue to advance, and you'll see that these super resolution techniques will be used in live cells in the near future.
Okay, it's past 9:30, and I think it's probably time to stop. Before we all go, and before we give a final thank you to all of our speakers, I wanted to mention that we have actually, the chemistry department is planning a display in their front lobby, which is going to honor 2 chemistry Nobel laureates from the University of Delaware. One of them you may remember is Dick Heck, who won the 2010 Nobel Prize, and there was a talk about his work in this series a couple of years ago. The other one is Dan Nathans, who was an undergraduate here, graduated in 1950. Remarkably enough, today is his birthday. He actually died in 1999, but he was born on October 30th in 1928. He won a Nobel Prize in physiology or medicine in 1978 for work on restriction enzymes. So the chemistry department is planning a display. This is a mock-up of it. When you walk into the front lobby of the building next year at this time, you should see something that looks sort of like that. We'll have pictures of each of these gentlemen on the wall there. So I wanted to mention that. I do hope you'll come back here next year. I'm planning to be on sabbatical, so somebody else will organize this. I will not do it for more than 8 years in a row, apparently. So, but I want to thank all of you for coming, and I want to thank again all of our speakers. Let's give them a round of applause.